Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on genetics, today we would be talking on polygenic inheritance, and for this very discussion we have once again with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Charu Dogra Rawat has immense experience and her teachings are very vivacious because she always believes in giving suitable examples so that it becomes easier for the students to get the in-depth knowledge on the topic. So dear friends, let's welcome our guest Dr. Charu Dogra Rawat and let's try to understand more and more about polygenic inheritance. Hello ma'am, welcome to Deviation lecture.
Thank you, Kritika. Good afternoon. So today we will be talking about polygenic inheritance, and as the name suggests, it's basically polygenic. So whatever the phenotypes which are governed by more than one genes are said to be polygenic traits, and these polygenic traits, how they are inherited from the parents, we'll be talking about that today. So the specific learning objectives would be to understand the basis of this quantitative inheritance or polygenic inheritance. We will see that polygenic is also called as quantitative inheritance. We'll understand the concept of additive alleles. We'll understand how these additive alleles leads to the continuous variation. We'll study some examples of polygenic inheritance and how to solve the numerical problems based on polygenic inheritance. I would also like to mention here that today's lecture mostly comes from Klug and Cummings' book Concepts of Genetics 11th edition. So that's the basic reference for today's lecture. So quantitative genetics, when we say it, is concerned with the inheritance of differences between individuals that are a matter of degree rather than kind. That is, they are quantitative in nature and they are not qualitative. For example, height, weight. When you study about these traits, we have to study about the degree of inheritance rather than whether they are inherited or not. The traits such as heights in humans, milk and meat production in cattle yield, and seed protein content in various crops cannot be assigned to distinct and separate categories as, for example, human blood type such as A, B, AB or O. Squash fruit shape was spherical, disc shaped or elongated. Or fruit fly examples which we have studied, for example, had eye color that was either red or white. So they were discrete traits. They can be defined or classified into different categories. But the traits such as height, there is a whole range of heights which is present. Or for example, the milk and meat production in cattle. So there can be a whole range of milk and meat production which could be there. So all these traits, and they are all measurable traits. You can measure the height, you can weigh the seed foliage which is produced. So everything basically relates to the quantitative inheritance, and these such are called as the quantitative traits. They show a kind of a continuous variation, as I say. So they show a range, they show a continuous variation from either the lighter to the darkest or lowest to the highest. So they show a kind of a continuous variation. The continuous variation across a range of phenotypes is measured and described in quantitative terms. The genetic phenomena is known as the quantitative inheritance. Now, because these varying phenotypes result from the input of genes at more than one and often many loci, they are said to be polygenic. So poly means many and genic means gene. So inheritance which is governed by a number of genes rather than probably one or two, more than one or two genes present at different loci, then they are termed as the polygenic inheritance. The quantitative traits whose phenotypes result from both gene action and environmental influences are often termed multifactorial or complex traits. So today we will also see that there is an environmental influence and we have to take into account this environmental impact which is causing the variation also. So till now we have been only talking about the genetic impact which basically governs a particular phenotype, but we will also see that the environment influence can also be there, and when certain traits which are governed both by the genotypic constitution as well as the environmental effect, they are said to be complex traits or multifactorial traits. Before we go on to talk about the quantitative traits, we'll talk about meristic traits. There are two different types of traits also. The meristic traits in which the phenotypes are described by whole numbers. For example, include the number of seeds in a pod or the number of eggs laid by a chicken in a year. These are all quantitative traits, but they do not have an infinite range of phenotypes. So when there are no infinite range of phenotypes, it is divided into whole numbers, either 1 or 2 or 3 or 5, then they are said to be meristic traits. For example, a pod may contain 2, 4 or 6 seeds, but it cannot have this 5.75. So there is a long range of variation, but it is not continuous. So discrete numbers are there, but there is a whole lot of variation which is there, and they are termed as the meristic traits. Then there are certain traits which are called as threshold traits. They are having a small number of discrete phenotype classes. The one example is type 2 diabetes, also known as adult onset diabetes, because it typically affects individuals who are middle-aged or older. The population can be divided into just 2 phenotypic classes for this trait. You know, the individuals who have type 2 diabetes and those who do not have type 2 diabetes. The combination of alleles present at multiple contributing loci give an individual a greater or lesser likelihood of developing this disease. These varying levels of liability form a continuous range. At one extreme, those at very low risk for type 2 diabetes, while at the other end of the distribution are those whose genotypes make it highly likely that they will develop this particular disease. So as shown in the graph, you can see that the number of predisposing alleles in genotype. So there are the lower ones where basically there are low risk people with type 2 diabetes, and these are where the high risk people, clinical diabetic condition is there, and usually they have a zone which is termed as the threshold zone, and therefore the threshold traits. So these are also two different kinds of traits. Apart from, you know, they are quantitative traits, but they do not show continuous variation, or there is a bias towards certain discrete phenotypic classes which is there. So now we will look at whether these quantitative traits now are inherited in a Mendelian fashion or not. So we have learnt about the laws of Mendel, and Mendel says that, you know, the genetic alleles basically they are segregated when the formation of gametes is there, and even if the genes are there, they are independently assorted, and therefore you get particular ratios in the F1 or in the F2 generation which you get. So looking at the quantitative traits, whether they are Mendelian or not, the geneticist William Bateson and G. Yule, adhering to a Mendelian explanation, proposed what is termed as the multiple factor or multiple gene hypothesis, in which many genes, each individually behaving in a Mendelian fashion, contribute to the phenotype in a cumulative or quantitative way. So Bateson and Yule basically proposed that quantitative traits are inherited, or the phenotype of the quantitative traits are because of the cumulative or quantitative or additive manner, and they propose this multiple factor hypothesis, which we will describe in detail in a while. The multiple gene hypothesis was initially based on a key set of experimental results published by this particular scientist Herman Nielsen L. in 1909. Nielsen L. used grain color in wheat to test the concept that the cumulative effects of alleles at multiple loci produce the range of phenotypes seen in the quantitative traits. So what experiment did he do was that he took wheat with red grain and crossed it with the wheat with white grain. Now F1 generation demonstrated an intermediate pink color, which at first sight suggested that incomplete dominance of 2 alleles at a single locus is there. So again, you have already studied about the deviation from Mendel's laws and the Mendel's laws, and if there is an incomplete dominance, then basically when both the red and the white gene allele is present, then it will show an intermediate pink color which is there. So when they crossed this red parent with a white parent, then they found the intermediate color, and by looking at this, this looks like an example of an incomplete dominance. However, in the F2 generation, Nielsen L. did not observe the typical segregation of a monohybrid cross. It did not. Approximately 15 out of 16 plants showed some degree of red grain color, while 1 out of 16 plants showed white green color. So otherwise it was, a typical, if you consider it as the segregation of the like happens in the monohybrid clause for both the alleles, then this ratio would not have come. But what they observed was that 15 out of 16 had some redness in there. Again, there was a variation, but some redness is there, and 1 out of 16 plants showed the white grain color which is there. The careful examination of the F2 revealed that grain with color could be classified into 4 different shades of red. Now, because the F2 ratio occurred in 16ths, it appears that 2 genes, each with 2 alleles, control the phenotype and they segregate independently from one another in a Mendelian fashion. So, the ratio, because the total number of progeny is basically the F2 ratio occurred in 16ths, which is the typical of a dihybrid cross, because now they are having 2 alleles as shown here, A and B. So, basically, and the F2 generation is 16th, the ratio is 16, therefore, it must be inherited in a Mendelian fashion, and they basically propose that each of the 2 alleles, they control the phenotype, but they segregate independently from one another in a Mendelian fashion. So, what is happening here? They gave this concept of the additive alleles. What is an additive allele? That each gene has one potential additive allele that contributes approximately equally to the red grain color, and one potential non-additive allele that fails to produce any red pigment. So, for example, in the parent now, both are homozygous. The red parent contains only the additive alleles. So, instead of, we are not calling them as dominant alleles, but now we are quantifying it. So, we are calling them the additive alleles. So, there are 4 capital, there are 2 capital A, 2 capital B. So, all the 4 alleles are said to be additive alleles. However, when the white color is there, it is said to be basically the non-additive alleles which are there, which is producing a white color. Now, looking at the F1 generation, the F1 plants would be heterozygous, capital A, small a, capital B, small b. It contains now additive as well as the non-additive alleles. So, 2 capitals are there and 2 small letters are there. So, it has 2 additive alleles and 2 non-additive alleles, and therefore, it expresses what is termed as the intermediate pink phenotype color. So, that explains the F1 generation. Now, what is happening in the F2 generation? Each of the F2 plants has 4, 3, 2, 1 or 0 additive allele. So, you can see the number of gametes which will be formed and when they will be combined. So, when these are the number of gametes that would be formed, and these are the genotypes of the individuals which would be there, and looking at them you can see that if they have all the 4 capitals, then they will have 4 additive alleles. So, this combination can be there. There can be a capital A, small a, capital B, the 2 capital A's, small capital B and a small b. So, 3 additive alleles are there. So, in a way, there in the F2 alleles present, there will be the entire range from 4, 3, 2 and 1 to 0 additive allele, where there will be only non-additive alleles which are present. So, while F2 plants with 4 additive represent one of the parent and the no additive allele represent the other parent which is there. Now, plants with this 3, 2 and 1 additive allele constitute the other 3 categories of red color observed in the F2 generation. So, additive means when they have the 4 additive alleles, then they have the most intensified color. When they are having no additive allele, which is 0 in number, then they are having the least effective red color or basically the white color, and in between, depending upon what additive allele is present, how many number of additive alleles are present, there is a whole range of colors that have occurred. So, that explains these 3 intermediate colors which are there. So, the greater the number of additive alleles in the genotype, the more intense the red color expressed in the phenotype, as each additive allele present contributes equally to the cumulative amount of pigment produced in the grain. So, now we are considering it as equally, although later on we will see that it is not an equal contribution, but probably when we talk about additive alleles and when we quantify it and put it into numbers, we will assume that they are contributing equally to the phenotype. So, the number of additive alleles will govern the occurrence of the phenotype from 2 major extremes of the parents, and then in between, depending upon how many number of additive alleles are present, you can have a range of different phenotypes which is there. Now, genetic ratios resulting from crossing two heterozygotes when polygenic inheritance is in operation with 1 to 5 gene pairs. So, you can look at this, when there is a 1 gene pair, there is a possibility of these are the individuals that will occur. So, if there are 2 gene pairs which we are talking about, then the polygenic inheritance is in operation, and there will be such a kind of a range of phenotypes which will be there. The more the number of genetic pairs we are taking into, or more gene pairs are governing a particular phenotype, more will be there will be the variation in the phenotype which can be observed. So, these are the number of individuals which are which is basically present, and this is the frequency occurring in a particular population. So, you can see that they are ranging from 1 to 1, and then they have intermediate numbers which is there. So, these are the gene pairs, and each phenotype results from a different number of additive alleles which is there. So, each phenotype, so there is a whole range of phenotype which is occurring. Now, these additive alleles explain the basis of, this is the basis of continuous variation that is reflected in the polygenic inheritance. So, the multiple gene hypothesis consists of the following major points. First is that the phenotypic traits showing continuous variation can be quantified by measuring, weighing, counting and so on. So, they can be, they are quantifiable, the phenotypic traits that show continuous variation. 2 or more gene loci, often scattered throughout the genome, account for the hereditary influence on the phenotype in an additive manner. Because many genes may be involved, inheritance of this type is termed as polygenic. Each gene locus may be occupied by either an additive allele that contributes to a constant amount to the phenotype, or a non-additive allele which does not contribute quantitatively to the phenotype. The contribution to the phenotype of each additive allele, though often small, is approximately equal. We now know that this is not true, but just for assumption that because additive allele is giving rise to the continuous variation of polygenic inheritance, we believe that they are contributing equally, which is there. And together these additive alleles contributing to a single quantitative character produce substantial phenotypic variation that is found. So, this is the points that explain the polygenic inheritance and explain the basis of the continuous variation found in polygenic inheritance to be the presence of these additive alleles which is there. Now, how do you calculate the number of polygenes that is governing a particular phenotype? If the ratio of F2 individuals resembling either of the two extreme P1 phenotypes can be determined, the number of polygenes involved may be calculated as 1 upon 4 raised to the power of n, which is equal to ratio of F2 individuals expressing either extreme phenotype. So, one has to look for the ratio of the parental a phenotype which is present in the F2 generation, and if that can be determined, the number of genes approximately governing that particular trait can be deduced. In the example of, for example, the red and white green color, in the wheat green color, 1 upon 16 of the progeny are either red or white like the P1 phenotypes. This ratio can be substituted on the right side on the equation and then calculated n. So, you can see that this red and white wheat grain color is basically governed by 2 of the genes which are present at different loci. These are the determination of the number of genes involved in a quantitative trait. So, you can see if only a single gene is governing, then there are 3 distinct phenotypic classes which can be present. Then if there are, which we have already seen that individuals expressing, so this is basically a monogenic inheritance, and we have seen that there is a 1:2:1 ratio which is there. If there are 2 genes which are present, then probably there are 5 distinct phenotypic classes which can be there. And if there are 5 phenotypic classes that are occurring, then there is a possibility that 2 genes are governing that particular phenotype. 7 distinct phenotypic classes, 3 polygenes, 9 phenotypic classes, 4 polygenes, so on and so forth can be determined. For low number of polygenes, it is sometimes easier to use the equation which is termed as 2n plus 1, which is equal to the number of distinct phenotypic categories observed. For example, when there are 2 polygenes involved, then 2n plus 1 is equal to 5, and each phenotype is the result of either 4, 3, 2, 1 or 0 additive alleles. If n is equal to 3, then 2n plus 1 is equal to, so that is how you can identify the genotypes that will occur. So the phenotype, the phenotypic result which is there. So if n is equal to 3, there are 3 polygenes which is governing a particular phenotype, then there will be 7 phenotypic classes that will result because of the 6, 5, 4, 3, 2, 1 and 0 additive alleles which is there. Now both of these are simple methods for estimating the number of polygenes involved in a quantitative trait, and they assume that all the relevant alleles contribute equally and additively, which is not usually the case, and the phenotypic expression in the F2 is not affected significantly by environmental factors. So when there is no environmental contribution, then they both the alleles or all the alleles are contributing additively and equally, then basically this is these methods can be used to estimate the number of particular polygenes involved in a quantitative trait. For many quantitative traits, however, these assumptions may not be true. Another example of polygenic inheritance is the skin color in humans. Skin color in humans is an example of polygenic inheritance pattern. The genes for skin color are located at a minimum of 3 chromosomal locations or loci. That is what experts initially found out. At each of these loci, the allele for dark skin is dominant over the allele for light skin. Therefore, a wide variety of skin colors is possible depending upon how many dark skin alleles are present. The number of total dark skin alleles from all the 3 genes determines the color. So these are the 3 genes, and on each genes there are 2 alleles which are present, and out of the 2 alleles, if it is a capital D that is dominant and a small d is recessive in this particular case. So when all the 3 genes are inherited, then they contribute the number of alleles which is there. For example, in this extreme case, there are all small d's which are inherited. There is no capital D is dominant, giving rise to the dark color, affecting the melanin production in the individual. And again, in this case, we are assuming that there is no environmental factor which is taking place. So we are not identifying that the location of the people where they are staying, where there is more sun, there is more UV. We are not considering the contribution of the environmental factors. All we are talking about the genes right now. So in this particular individual, because there is no capital D that is present, the light alleles there is 6, and for dark allele there is 0, so it is lightest in color. In the dark one, there are 6 alleles which are capital D, and there is no allele which is a small d, so all the 6 are dark alleles, and therefore he is darkest in color. And in between there is a variation depending upon whether there is a 1, 2, 3, 4 or 5 additive dominant capital D alleles are present or not. It can also be shown with the binomial expansion, which is, for example, a plus b raised to the power of 6, where the letter a is the number of capital letters and the letter b represent the number of small case letters which is there. So if you can see, then the binomial expansion leads to this particular equation, and in this case there is the exponent depicts the number of dominant alleles which is present, and the coefficient represents the number of offspring which would be there. So there will be 6 offspring which will have a capital 5 allele. So the ratio of the phenotype type, number of offsprings which will be there. So they will have capital D and so on and so forth. So that is can also be reduced by the binomial expansion. So inheritance, if we look at that, for example, if you are crossing a very dark individual consisting of all the capital A, B and C, so all the 3 loci there is a capital. Now we are not representing it only by D, but because there are 3 different genes, we are representing them by A, B and C here, but their capital means that they are a dominant allele, dominant color allele, and this is a recessive color allele. F1 generation will be heterozygotes, and looking at the number of gametes, you will see there will be a whole variation of colors which will be there, and it will give rise to a normal distribution that can be seen. From one end there will be the lightest in color, and on the other hand there will be the darkest in color, giving rise to all the range in between. Currently, the research suggests that variants in more than 20 known genes determine human skin color and not just the 3 genes. Their products are nearly all involved in stages of melanin production. Some variants specified by a particular gene encourage the production of more melanin. Others switch the ratio of U to pheomelanin or alter the packaging of melanin into melanosomes. So, inheritance of skin color in humans is a complex trait, and that's the basic thing. Now we will quickly look at some of the problems based on the polygenic inheritance. Now that how can you solve when you go in for some competitive exams or when you try to solve these genetic problems. For example, assume that a height in a plant is controlled by 2 gene pairs and that each additive allele contributes 10 centimeter to a base height of 10 centimeter. So the base height means that when there is no additive allele that is present, the height will be 10 centimeters. So you have to find out what is the height of a capital A, capital B plant, and so predict the phenotypic ratios of F1 and F2 plants in a cross between small a, small b, and capital A, capital S, capital B, and then list all the genotypes that give rise to the plants that are 20 and 40 centimeter in height. So let's look at the first one. The height of a capital A, capital B plant would be 50 centimeter because each additive allele in here is caps, and it is an additive allele and it is giving 10 centimeters, it is contributing 10 centimeter to the height. So 4 additive alleles, so 4 into 10 is 40 centimeters, and the base height was 10 centimeters, and therefore it will be of 50 centimeters in height. Predict the phenotypic ratios of F1 and F2 plants in a cross between these two. So the F1 plants will be 30 centimeters tall and the genotype will be a heterozygote. So basically the genotype is a heterozygote. It will occur, that is there will be 2 additive alleles. So 2 additive alleles means 10 plus 10, 20 centimeters plus 1 base height, which is again 10 centimeters. So it will be 30 centimeters tall. F2, there will be a range 10, 20, 30, 40 and 50 centimeter plants in the ratio 1, 4, 6, 4:1 ratio. This answer can be calculated from the binomial expansion x plus 1 raised to the power of 2n is equal to 1. Here I mention x plus y and not a plus b, just not to confuse with these a and b alleles which is there. So x is a non-additive allele, y is an additive allele, and n is the number of genes. 2 is expanding the equation. Number of genes which is equal to 2. So it will be 2 here, and expanding the equation you will get x raised to the power of 4 plus 4x square cube y, 6x square y square. It's a typical binomial expression which is there. The exponent I told you represent the number of alleles which will be there, number of that particular. So x, 4 number of additive alleles, in this case 3 number of additive allele and 1 non-additive allele y. And the coefficient tell you about the ratio of the phenotype, the number of offsprings of the phenotype is which are there. Keeping in mind the exponents reflect the number of those alleles in that genotype, what we get coefficient reflect the proportions, we get the distribution of plant. So what you will get 10 plus 10 plus 10 plus 10 which is 40 centimeter plus base height which is 50 centimeter. So you will get one individual or ratio of one phenotype as 50 centimeters. In this you will get 4 offsprings in 4 in ratio which will be containing 3x which is 30 centimeter plus 10 centimeter and plus 10 centimeter which is again 50 centimeter which will be there. So on and so forth, you will have all the variety because in this case there will be only 10 centimeters which is there. So all the recessive alleles are there. So this particular case will give you only 10 centimeter which is there. The third thing is list all the genotypes that give rise to plants that are 20 centimeter and 40 centimeter in height. So 20 centimeter plants will have only one additive allele because the base height is 10 centimeter and another 10 centimeter is contributed by the additive allele. So the genotype would either be a capital A, only one capital would be here, either A or B. So that will be the genotype. For 40 centimeter plants, 2 additive alleles can be there. So it can be a capital, 3 additive alleles would be there because the base height is 10 centimeter and then 10 plus 10 plus 10. So 3 additive alleles, the genotype could be capital A, capital B, small b, or capital A, small a, capital B, capital B. So these are the 2 genotypes which are there. Let's look at another example. In a cross where 3 gene pairs determine weight in squash, what proportion of individuals from the cross this and this will contain only 2 additive alleles, and what genotype fall into this category. So let's look at the number of gametes they will produce. So the gametes produced in this particular cross will be these lying on the row and column here, and then looking at the combinations that will arise. The parentheses tell you about the number of offspring which would be there. The cross is between this and this, and as seen in the diagram, 1/8 of the progeny will have 2 additive alleles, which is capital A and capital C. The genotype of this class will be this one, which is represented. So the number of additive alleles are shown in the in each case in the parenthesis. So 1 out of 8 basically will have the 2 additive alleles which are there. So all the 5 additive alleles or 2 additive alleles. So that kind of region, the phenotypes can exist. Another example which we can take, an inbred strain of plants has a mean height of 24 centimeters. A second strain of the same species from a different country also has a mean height of 24 centimeter. The F1 plants from a cross between these two strains are also 24 centimeter high. However, the F2 generation shows a wide range of heights. The majority are like the P1 and F1 plants, but approximately 4 out of 1000 are only 12 centimeter high. So, you know, half the height, and 4 out of thousands are 36 centimeters in height, another 12 added to the median height. So, there is a continuous variation. So, there is a plant which you are getting for 12 centimeter height, there are plants which you are getting for 24 centimeter height, and there are plants which you are getting for 36 centimeter height, and there is a whole variation, and the number of offsprings are also given. So, the first thing you have to find out is how many gene pairs are involved, and as I told you, for calculating the gene pair, it is 1 upon 4 raised to the power of n is equal to the number of parental offsprings which you can observe. The frequency of either extreme phenotype gives us the n, the number of gene pairs, the frequency of one extreme phenotype 1 upon 4 raised to the power of n is equal to 1 upon 250, which is there because 4 divided by 1000 individuals gives rise to 1 upon 250. Therefore, the number of gene pairs is basically 4. So, you know, 4 into 4 into 4 into 4 gives 256. So, approximately 4 genes will be controlling this particular trait. The second is how much does each gene contribute to the plant height. Now, the maximum contribution of additive alleles is 24 centimeters. Since 8 additive alleles contribute 24 centimeter, each additive allele contribute 3 centimeter, which is there. We already know that the mean height is 24 centimeter and the maximum contribution is basically the 8 additive allele, because that's the highest one. You know, the mean is the highest one, and there is a range from lower to the highest which is there. The next is indicate one possible set of genotypes of the parent as well as the F1 plants that could explain these particular heights. Each parent has now 4 additive alleles. Since the F1 also have 4 additive alleles, the parents must each be homozygous. The additive alleles of one parent are not present in the other. For example, the genotypes could be capital A, capital B, 4 additive alleles are there, but this will not be present in the other one which is there. Other genotype following such patterns could be there. So, you know, there can be capital A, capital C, and small ones, the rest of them. So, such kind of genotypes would be present for the parent which is there. The last is indicate one possible set of genotypes to account for F2 plants that are either 18 centimeter or 13 centimeter in height. An 18 centimeter plant has 2 additive alleles. Any genotype such as this or this, you know, when only 2 additive alleles are there would work. A 33 centimeter plant has 7 additive alleles, and any genotype representing as such as this and this would also work. So, you basically have to calculate that for having 33 centimeter, how many additive alleles do you need? So, you know that, you know, each additive allele is contributing 3 centimeter. So, for 33 centimeter, you at least need to have 7 additive alleles, and therefore, these things can be, you know, these genotypes can be deduced where you will have 7 of the caps and the rest can be 1. And other genotypes in the possibility of another genotype is that there can be one small a, there can be one small b, or there can be one small c, and such things can occur in this particular case. Thank you. How are polygenic traits studied? In this particular session, we will talk about the statistics or the statistical terms which are used for calculating the, for analyzing the quantitative character of a particular trait, and we will also look at how it is done in the human. So, the particular learning objectives would be to understand the statistical terms for studying polygenic traits, to understand how analysis of quantitative characters are done based on these statistical measures, to understand the concept of heritability, and to understand how this heritability is used for artificial selection, and to understand the estimation of heritability in humans. And again, I would like to say that most of the major part of this particular presentation comes from the book of Klug and Cummings, Concept of Genetics, 11th edition, which is there. Now, it is not usually feasible to measure expression of a polygenic trait in every individual in a population. So, a random subset of individuals is usually selected for measurement to provide a sample. Now, if the sample measured for expression of a quantitative trait is sufficiently large and also representative of the population from which it is drawn, we often find that the data forms a normal distribution. That is, they produce a characteristic bell-shaped curve when plotted as a frequency histogram which is there. So, the continuous variation give rise to such a kind of a normal distribution which is there. For this normal distribution, certain statistical terms or statistical measures are computed. One is the mean. The mean provides information about where the central point lies along a range of measurements for a quantitative trait. The measurements tend to cluster around a central value. This clustering is called as the central tendency, and the central point is termed as the mean. Specifically, the mean is the arithmetic average of a set of measurements and is calculated by the mean is equal to sigma xi divided by n, where x bar is the mean, sigma xi represents the sum of all individual values in the sample, and n is the number of individual values which is there. The mean provides a useful descriptive summary of the sample, but it tells us nothing about the range or spread of the data. So, the central cluster or the average of two different sets could be the same, but how much lower it is going or how much higher it is going, or what is basically the spread of this particular graph, it does not reveals that. It only reveals that this is the maximum or this is the average data in the particular set or average measure in the particular set which is there. So, for that, a second statistic called as the variance that provides information about the spread of data around the mean is also calculated. The variance or S square for a sample is the average square distance of all measurements from the mean. It is calculated as S square is equal to sigma xi minus x bar whole square divided by n minus 1, where the sum of the square differences between each measured value and the mean is divided by 1 less than the total sample size. So, that will tell about the spread, the distance from the mean which they are getting. So, all the values which are how much far they are from the distance, and when you square that distance, divide them by the sample size n minus 1, you get the variance. It is possible for the two sets of sample measurement for a quantitative trait to have a same mean but a different distribution of value. So, in here, as I was mentioning, you can see that these two particular graphs have the same mean, but the spread around the mean is different. This range will be reflected in the different variances. The estimation of variance can be useful in determining the degree of genetic control of traits when the immediate environment also influences the phenotype. So, this variance is quite important for knowing how much the environment is also influencing. So, the degree of genetic control, we will see that in a while. Another term which is used is called as the standard deviation. Now, because the variance, it is a squared value, its unit of measurement is also squared, for example meter square or gram square. Now to express the variation around the mean in the original units of measurement, we can use the square root of the variance, a term which is called as the standard deviation. So S is equal to square root of S square, and you can see that sample inclusion for various S values, if you give, and you know how much spread does it cover. If the value can be around 1s of the mean plus and minus, then it includes the 68.3 sample which is included. If the mean is 1.96 s around the mean, then 95% of the sample is covered here. So it will be more, if we can look at the normal distribution curve which we can see. So this is the mean which is there. So plus minus 1 will cover the maximum amount of graph which is there. So it will cover the 60%, and plus minus 1s. So this will be plus 1s, this will be minus 1s, and it will cover the 95% of the the bell-shaped distribution which is there. So that is how the standard deviation is reduced. So 3s will cover 99.7s. So the percentage of individual values within a normal distribution that fall within different multiples of the standard deviation. The value that fall within one standard deviation to either side of the mean represents 68% of all values in the sample. More than 95% of all values are found within 2 standard deviations to either side of the mean. This means that the standard deviation can also be interpreted in the form of probability. For example, a sample measurement picked at random has 68% probability of falling within the range of one standard deviation which is there. Another term which is used is called as the standard error of mean. If multiple samples are taken from a population and measured for the same quantitative trait, we might find that their means vary. Theoretically larger, truly random samples will represent the population more accurately, and their means will be closer to each other. To measure the accuracy of the sample mean, we use the standard error of the mean, which is calculated as capital S x bar, which is equal to S divided by square root of n, where S is the standard deviation and square root of n is the square root of the sample size. Because the standard error of the mean is computed by dividing S by square root of n, it is always a smaller value than the standard deviation. So standard error of mean is always a smaller value than the standard deviation. Another term which is used is called as covariance. The covariance statistic measures how much variation is common to 2 quantitative traits. It is calculated by taking the deviations from the mean for each trait, because for example, sometimes what we do is we do not study one trait independently, but we study a number of traits or 2 traits simultaneously, and then we use the statistical term covariance. This given a pair of value for each individual. The 2 values are multiplied together and the sum of all these individual product is then divided by 1 fewer than the number of samples. Thus the covariance of 2 sets of trait measurements x and y is calculated by this particular formula which is there. So let's look at the analysis of a quantitative character. To apply these statistical concepts, let's consider a genetic experiment that crossed 2 different homozygous varieties of tomato. One of the tomato varieties produced fruit averaging 18 O's in weight, whereas fruit from the other averages 6 O's. The F1 obtained by crossing these 2 varieties has fruit weights ranging from 10 to 14 O's. F2 population contain individuals that produce fruit ranging from 6 to 18 O's. So from 6 to 18 O's which is there. In the F1 generation there were 10 to 14 O's which was there, and this is the frequency number of individuals which are found. So one of the tomato produces fruit averaging 18 O's in the weight. So other average is 6 O's. So that was a distribution which is there. The mean value for the fruit weight in the F1 generation can be calculated as given by the formula sigma xi divided by n. So that will be 12.04. The mean value for fruit weight in the F2 generation is calculated at 12.11. So you can see, the spread is different in F1 and F2 generation, but mean is coming to be similar. Mean values are similar. The range of variation can be quantified as the sample variance as the sum of the square differences by this particular formula. Now, the variance is found to be 1.29 for the F1 generation and 4.27 for the F1 generation. When converted into standard deviation, the value becomes 1.13 and 2.06. Therefore, the distribution of tomato weight in the F1 generation can be described as 12.04 plus minus 1.13, and in the F2 generation it can be described as 12.11 plus minus 2.06, which is there. Assuming that both tomato varieties are homozygous at the loci of interest and that the alleles controlling fruit weight act additively, we can estimate the number of polygenes involved in this trait. Since 1 divided by 72 of the F2 offsprings have a phenotype that overlaps one of the parental strains. So, 72 total F2 offsprings are there, and 1 weighs 6 O's and 1 weighs 18 O's. So, we can calculate that 1 divided by 4 raised to the power of n, it should be divided by and not greater than 1 divided by 4 raised to the power of n is equal to 1 divided by 72, which indicates that the number of genes which is controlling the weight of the tomato is basically between 3 and 4, providing evidence of the number of genes that control the fruit weight in these tomato strains. So, that is how you analyze a quantitative character by using the statistical terms such as mean, variance, standard deviation, etc. The covariance can then be standardized as yet another statistics which is called as the correlation coefficient. The calculation of R is given by covariance xy divided by Sx(xy), which is there, where Sx is the standard deviation of the first set of quantitative measurements and Sy is the standard deviation of the second set of quantitative measurements. The values for the correlation coefficient can range from 1 to +1. Positive R value means that an increase in measurement for one trait tends to be associated with an increase in measurement for the other trait. So, covariance we were talking about when we were comparing two traits and we were looking at the variance from the mean in these two particular traits which are there. Then based upon these covariances, we can find upon the correlation coefficient that whether one trait is influencing the other or not. So, the measurement so increase in the measurement for one trait is associated with the increase in the measurement, not influence but associated. The genes are not affecting each other, but we are just looking at whether the measurements which have been obtained, then two different measurements, whether they are associated or not. While negative R values means that increase in one trait are associated with the decreases in another. One important point to note about the correlation coefficient is that even significant R values close to +1 or -1 do not prove that a cause and effect relationship exists between these two traits. Correlation analysis simply tells us the extent to which variation in one quantitative trait is associated with variation in another, not what causes the variation. So, as I said, it is not explaining the effect of the variation, or it is not explaining the influence of one gene onto the other. All it depicts is the association that whether the increase in one of the traits or whether the phenotype of a particular trait is associated with the phenotype. So, there is no association between the cause of the variance which is occurring. We cannot say that the height is increasing and therefore the weight is also increasing, but we can observe that there is a correlation that whenever the height is increasing, the weight is also increasing. So, such kind of a correlation. So, one should always keep that in mind, that it is not that both might have the same cause, or it is not explaining that the cause of the variance in the data, but just correlating these two particular traits in terms of the phenotypes that they are producing. Now, we come to one important term which is termed as heritability, that is describe what proportion of total phenotypic variation in a population is due to genetic factors. So, till now we were talking only about the genetic constitution and genotypic, the genes which are affecting a particular phenotype. So, genetic contribution is, and then we talked about the quantitative trait where many genes are influencing a particular phenotype and they are adding, they are doing so in terms of cumulative or additive manner. They are doing so in cumulative manner. But there is an environmental influence also, and this the proportion of total phenotypic variation in a population due to genetic factors is termed the heritability. Rest is given the name of a environmental influence which can be there. Now, for a multifactorial trait or a quantitative trait in a given population, a high heritability estimate indicates that much of the variation can be attributed to the genetic factors, with the environment having less impact or expression of the trait. With the low heritability estimate, environmental factors are likely to have a greater impact on phenotypic variation within the population. So, a heritability estimate tells us the proportion of phenotypic variation that can be attributed to genetic variation within a certain population in a particular environment. If we measure heritability for the same trait among different populations in a range of environments, we frequently find that the calculated heritability values have large standard errors. This is an important point to remember when considering heritability estimates for traits in human populations. A mean heritability estimate of 0.65 for human height does not mean that your height is 65% due to your genes, but rather that in the population sampled, on average, 65% of the overall variation in height could be explained by genotypic differences among individuals. So, heritability is a measurement or is an estimate which is generally of a population. So, we take a sample of a population from a population and derive the heritability, in which we can say that that percentage can be contributed to the genetic factors. As they say that the measure of the heritability of the same trait among different population and different environment usually have a very higher standard of standard errors which are there. So, we should not associate the heritability estimate with the contribution of genes as such, but within different environments when we are constituting, we say that so much percentage of heritability means that so much on average this much of overall variation is contributed by the genetic factors which is there. Now, heritability estimates are obtained by using different experimental and statistical techniques to partition VP, which is the phenotypic variation, into genotypic variance and environmental variance components which is there. So, genotypic variance is referred to as capital V subscript G and environmental variance at VE components. The third component of phenotypic variation is also there, which is genotype by environment interaction variance which is there. So, this VG into E, although it is a very small influencing factor, but it is there. Therefore, we have to include that also. So, for example, differences in yield between two wheat varieties at different soil fertility levels if we see. So, if there is no GE interaction, then the varieties show genetic differences in yield but respond equally to increasing soil fertility. So, when we are only talking about genotypic variance and environmental variance, then there are two different varieties. They have a particular genotypic variance, but that genotypic variance will be constant, and environmental variance will have the same effect on both the varieties. So, as the soil fertility will increase for both the varieties, the yield will increase in a similar manner, but this does not happen. And what shows that there is a G and to E interaction? Then variety A outyields B at low soil fertility, but B yields more than A at high fertility levels. So, this difference in the yield indicates that there is a genotype into E interaction which is present, and therefore, we also have to take this particular variance in between. If this variance was not there and there will be only genotypic and environmental variance which will be there. So, if the environmental variance is increasing, the genotypic variance will also increase, and both of them will show the parallel lines. But since they do not show it, that means there is a genotypic and genotype by environment interaction which is occurring. We will discuss about these two terms now. One is called as the broad sense heritability. It is represented by the term capital H square. It measures the contribution of the genotypic variance to the total phenotypic variance which is there. So, H square will be equal to Vg divided by Vp. So, what proportion of the phenotypic variance is genotypic variance gives the broad sense heritability. Heritability values for a trait in a population range from 0 to 1. A value approaching 1 indicates that the environmental conditions have little impact on. So, if this Vg is coming equal to Vp, then H square will be equal to 1, and therefore there is less of environmental influence which is there. So what we are talking about here is that the Vp will be equal to Vg plus Ve plus Vg into E. So considering Vg into E to be negligible, Vp constitute Vg and Ve. So Vg divided by Vp is the heritability component. So if Ve is coming closer to Vp, then this particular, the entire thing will be a genotypic contribution which is there, and therefore little impact of the environment which will be there. Low values close to 0.0 indicate the environmental factors, not genotypic differences, and largely responsible for the observed phenotypic variation within the population studied. Few quantitative traits have very high or very low heritability estimates, suggesting that both genes and environment play a part in the expression of most phenotypes for the trait. Vg used in the broad sense heritability, it should be noted that when we are talking about genotypic variance, the genotypic variance includes all types of genetic variation in the population. It does not distinguish between the quantitative trait loci with alleles acting additively as opposed to those for example epistatic or dominance effect. So it is taking these additive and dominance effects also into the Vg component. Broad sense heritability estimates also assume that the genotype by environmental variance component is negligible. These limitations mean this kind of heritability is not very useful in breeding programs. So usually you calculate this heritability for doing the breeding programs for selecting a particular kind of a trait. So if this particular kind of trait is not influenced by the genotype, or you are not sure whether the additive alleles is the basis of the inheritance of this thing, but there can be a dominance or there can be an epigenesis, then you cannot predict the outcome of a breeding thing or heritability of a advantageous trait which can be there. So the animal or plant breeders wishing to develop improved strains of livestock need more precise heritability estimates, and therefore they use a term which is called as the narrow sense heritability. Narrow sense heritability is a proportion of phenotype variance due to additive genotypic variance alone. So Vg is equal to Va plus Vd plus Vi, where Va is the additive allele variance, Vd is the dominance variance, and Vi is the interactive variance from the additive components that occur when two or more loci have epistatically, but this is almost negligible. So again, for making it simpler, we will ignore this and we will take Vg to be the sum of Va plus Vd which is there. And a narrow sense heritability estimate based on the portion only of the additive allele. So small h square will be Va by Vp. Obtaining Vi and estimating this, it will give rise to this. Heritability estimates are used in animal and plant breeding to indicate the potential response of a population to artificial selection for a quantitative trait. Narrow sense provides a more accurate prediction of selection response than broad sense heritability, and therefore it is widely used by the breeder. Heritability estimates are used, as we said, in animal and plant breeding to indicate the potential response of a population. For artificial selection, it is the process of choosing specific individuals with preferred phenotypes from an initially heterogeneous population for future breeding processes. Many traits of economic importance in crops and livestock, such as grain yield in plants, wheat grain or milk yield in cattle, and speed or stamina in horses, are polygenic and frequently multifactorial. Artificial selection for such trait is slower and more complex. So narrow sense heritability is used to predict the impact of selection. The higher the estimated value of H, the more likely the breeder will observe a change in phenotypic range for the trait. Partitioning the genetic variance components to calculate H square and predict response to selection is a complex task requiring careful experimental design and analysis. The simplest approach is to select individuals with superior phenotypes for the desired quantitative trait from a heterogeneous population and breed offspring from those individuals. The mean score for the trait of those offsprings can then be compared to that of the original population's mean score and the selected individual used as parents. The relationship between these means give the heritability. So the relationship between the mean score of a particular trait which we are choosing at is basically can be found out. This equation can be further simplified by defining M2 minus M as the selection response the degree of response to mating the selected parents, and m1 minus m as the selection differential, the difference between the mean for with the whole population and the mean for the selected population. So, h square reflects the ratio of the response observed to the total response possible, which is there. So, h square is equal to r by s, a narrow sense heritability value obtained in this way by selective breeding and measuring the response in the offspring is referred to an estimate of the realized heritability which is there. So, I would like to end here, and we will continue with more of this in our next session.
Definitely, we would be continuing later in the another session. So, dear friends, keep watching us and keep giving your feedback for this particular lecture. You can mail us at info.cc@nic.in. So, we would be meeting again. Till then, take care. Goodbye. Thank you, ma'am. Thank you so very much.